Okay, so let's move to the second part of our presentation this morning. And Yasser is proposing a research program dedicated to empirical and mechanistic brain modeling for characterizing patient-specific disease progression and implementing precision medicine. So, Yasser.
Thank you, Habib. Hi, everyone. In this second presentation, I will enter more on disease mechanistic modeling, considering not only macroscopic brain information, but also molecular data, and how we can use this modeling to identify, to understand better the disorder, but also to identify personalized treatment needs. As I mentioned before, our lab focuses on an integrative a perspective that try to doesn't consider only separate elements, but more how the different elements interact. And this includes multi-omics molecular information, which could be DNA methylation, transcriptomics, proteomics, metabolomics, with multimodal neuroimaging, which could be PET imaging or MRI, analysis of brain connections, behavioral data, lifestyle, behavioral, clinical information, in order to get disease long or representation of disease mechanisms at the individual level, and how we could use that information, those representation, to identify the effective treatments. You probably have heard a lot about the need to identify biomarkers in when we study neurodegenerative studies, and that's actually something that is very common. Let's say if you go to any Mayo conference in Alzheimer about Alzheimer or Parkinson or schizophrenia, any disorder, you will hear a lot the word biomarkers, and you also will hear a lot about the need to finding an optimal biomarker or a unique biomarker or 1-2 biomarkers that will be perfect to track disease progression and to know who will respond or not to a treatment. For me, that's kind of a fallacy. The more we study these diseases, the more we extend our view to a multifactorial view, the more we accept the complexity of neurological disorders, the more we understand that trying to identify a unique biomarker, a unique signal that could represent very well where a person, or predict where a person is going in terms of diagnosis or response to treatment is not entirely possible. So because it's like, we could see like the difference between fishing with a stick and a net, right? So like trying to find only one is fishing with one stick. It's going to a super vast sea and trying to, with only one biological factor, with only one dimension, solve all the multidimensional problem. And I will show how we could use data or try to integrate data coming from different modalities to respond to questions about disease advance and therapeutic needs. And we have to think too that if we have been, as a community, recognizing more and more the need to use data to track neurological disorders, we with time have also more and more data. So we have data coming from the molecular modalities, data coming from the imaging modalities, from the digital weighting modalities, from the new ways of making clinical evaluation, etc. At a point that we have millions of potential biomarkers to analyze. So what do? We go one by one, testing each of them, or we try to find ways to accelerate the testing and integration of all of them into unique composite scores or composite or integrated methods that would accelerate the whole process. And that's exactly what computational disease progression modeling techniques do, right? Is to, instead of focusing on separate biological markers and testing them, for 10 years or 5 years and spending a lot of time and energy on only one that might fail or might work to some extent, but will fail to capture the high heterogeneity of the disorders, we could then, instead we can use disease progression models that are mainly divided into major categories, so the empirical category and the mechanistic category. Empirical category are those disease progression models that are mainly focused on finding associations, correlations, or even making predictions, but that don't offer a biological explanation of a given disease. And I would say most of the research done so far have been using empirical models. So even like when we see, when we think in ApoE as the major disease genetic risk factor for Alzheimer's, that's based on, to some extent, on empirical analysis, right? Or when we do Y association studies, so that's just a regression of with a lot of genes, of the data coming and reflecting information about a lot of genes, and to explain clinical diagnosis. Or when we do correlations to find which biological factors associate the most with a given disease condition, that's still empirical. So at the end, at these models in which, yes, we can make predictions, or sometimes these predictions can be even very high. Like we can predict to some extent, like with a 90% of accuracy or more, who is an Alzheimer's patient compared with controls, or who we convert or not. Well, that can be very useful for the clinic under some conditions, but that is not useful to understand the disease mechanisms, right? On the other hand, mechanistic models are, as the word says, techniques of modeling that try to find explanations of why someone is developing a given disorder. Like when we assume, let's say, how genes interact and what is the cascade of genes interacting and proteins interacting to conduce to a given set of clinical characteristic or alterations. And so those modelings are more ambitious in terms of understanding the process, the whole process, and they are not so focused, or not necessarily that focused on making predictions. Why? Although they can, but the predictions might have lower accuracy than empirical. And I don't think that one type is better than the other one. I think at the end, for some type of applications, we need empirical modeling. For other types, or for other questions, for other, let's say, points or strategies, to define our strategies, we need a mechanistic modeling. And in fact, we can combine both to use the advantage of both types of modeling for improving the conditions of the person that are developing disorder. So I will cover some of the work that we have done using empirical modeling and some of the work that we have done using mechanistic, and how we can integrate those towards something more informative. First, I will start with some big data integration using machine learning. Specifically, as I mentioned before, one of the major challenges that we have to study progressive disorders, and in particular neurodegenerative disorders, is that they take decades to develop, right? So normally what we have done, and historically we have done, we take patients which already have Alzheimer's or Parkinson, and we compare them with the controls, but we miss what happened in the middle on the time of in the period that the person was developing the disorder. So we miss the initial triggering events. We miss what was happening before having the clinical symptoms. And in some way, those are the interesting, and are those periods which include probably the most relevant biological alterations, or the most relevant biological interaction that conduce to the disease, and also the periods that could have more relevant information for stopping the disease progression. So in order to infer from current existing data the timeline of the disease, we obtained the gene expression data of transcriptomics from postmortem brain from the ROSMAP database. It's a major study done in the States for about 1,200 participants from ROSMAP. So these are subjects that are accepted to donate their brain when they die, and then the brain, after this study, the ROSMAP have is probably the major brain bank of the world, and have characterized Alzheimer's extensively in terms of at the molecular, neuropathological level, clinical level. We also have gene expression coming from the blood from about 700 participants from the Alzheimer neuroimaging study. These are subjects that are alive. And then what we have here in the middle is a representation of the data of just the transcriptomic data, right? We have the rows are the subjects and the columns are the features of the expression of specific genes or transcripts. This is a lot of data normally. So we have like 4,000 features. So how, instead of going one by one, analyzing which ones are the ones that correlate the most with disease progression, what we did was we used a specific dimensionality reduction techniques, and particularly constructed PCA, that is a dimensionality reduction technique that try to find enriched patterns on your disease population relative to your background. It's like the background population, let's say the controls that haven't developed the disease yet, are like noise that is used to identify which components are more accentuated or are more informative in terms of disease progression. And then we assume that these components, let's say we finish with from 5 to 8 components, representing or summarizing the biological change at the transcriptomic level in the disease population. We assume that these components will allow us to represent each patient or participant in this case in a multidimensional space in which the position of the person in that space will be reflective of how close or far or how early or advanced is that person developing a given disease. Because these were constructed components, so proximity to the origin of that space will be reflecting or be proportional to how normal is a person, or how cognitively unimpaired is a person in terms of developing neurodegeneration. And the farther is someone from that space, will provide an idea of how far is that person in terms of how advanced is that person in terms of developing the disease. So then what we did was just to connect the dots. So if you want to measure how close or far is someone from the origin, all what you need to do is to calculate the geodesic distance in that space to the origin. So, and then that allows to define or to have a number that in this case we call pseudo time that reflects or give an idea of how advanced is someone in the timeline of the disease process. If you have thousands of brains, so you can assume, particularly for this kind of study, we know that some of the persons in those studies were, at the time that they were evaluated, they were at the early phase, others were at very advanced phases. So at the end, what we tried to do was to reorder this cross-sectional information that are snapshots, to reorder them in a way that they give an idea of the whole somehow movie, right? That will be reflecting or showing how the disease developed. So this pseudo time, when we observe it, or we try to analyze how it will be, if it would be really reflecting disease progression or not. We observed that it was significantly predicting the level of neuropathological evaluation in the brain. And this is molecular data, right? So what we are using here is molecular data, gene expression data, which could be from the blood or from the brain. And then we see that if we reorder that data, if we find a way to organize them that corresponds to the way that they were getting altered during disease progression, it reflects very well the level of neuropathological accumulation in the brain. And it also reflects memory decline and executive function decline. So it reflects the cognitive impairment that happens in the subjects. This is not a mechanistic model. However, because we have ways associated to the different genes, we can always go back and analyze which genes were the most predictive of the disease progression, or essentially which genes allowed to or contributed to position the persons closer to normality, or to non-normality, like to non-cognitive impairment, or to disease, advancing disease in that timeline. And then we can interrogate those genes. Which specific molecular functions are the most predictive genes associated with, which specific cell types they are associated with. So although it's not a mechanistic model, it's an empirical model, you finish having a pool of disease mechanisms that are not necessarily associated with neuropathology accumulation, that are informative of the whole disease progression process. So we verify that this pseudotime or disease progression score that we get just from blood sample, for example, is predictive of tau accumulation or amyloid accumulation in the brain. Is more predictive of the comorbidity. So it predicts better the combination of accumulation of different neuropathology, that each of them separately. And importantly, it changed across the different clinical states. And what was most relevant for us was that it was significantly different, and we can see that in the bottom graph on the right. It was significantly different for the persons that were converting versus those that compared with those that were not converting. So it could be in the clinic. It could be used as a predictor, minimal invasive predictor for converting from one clinical state to a more advanced clinical state in Alzheimer's disease. Right. So it can have a useful clinical application. Then we surprisingly, or nicely, we found that 90% of the most predictive molecular pathways identified in the brain that contributed to create or to the estimation of the pseudotime of subjects that were dead were also identified as top predictors in the blood, which highlights the importance of the peripheral brain gases. And these were obtained from different databases. Even when we use post-mortem data from one database and in vivo data from another database, the molecular pathways that the model is suggesting are relevant and more relevant to predict the progression of Alzheimer's disease were common or highly common between the brain and the peripheral blood data. And this includes B-cell activation. Now we know that how important is immune response for the development of neurodegenerative disorders, in particular for Alzheimer's. It also includes angiogenesis, that is involved in the generation of new vessels, or everything related with the vascular system and how it regenerates. Also P53, which is related to the cell cycle with angiogenesis, signal transduction, blood coagulation. So some of them were related to myophotoproteins, but as we can see, the top of them were also related to immune system, vascular system, so other biological processes that not necessarily directly related to myophotoproteins accumulation. That was a good, I would say, a good way to go from molecular data to an understanding of first to reorganize the subjects in a way that we get a better, more continuous picture of how the disease developed across the whole disease spectrum. It also was a relatively good way to get in vivo an integrated, aggregated in vivo marker or disease score that predicts if a person will develop the disease or not. But at the end, RNA is only one of the multiple layers of the multiple, say, entities or biological entities that contributes to our body and brain's functioning, right? So we also have the other layers like methylation, proteins, metabolites. So all of them interact. So one of our main purpose is not to keep working with only one data or level at any specific level or data modality, right? Because we know that at the end what happens with brain function is something that results from interactions between different layers of different entities, biological entities. So in order to consider other type of biological levels, then we expanded the previous model to now include epigenetics, transcriptomics, proteomics, and metabolomics. Again, We have data from the brain and data from the blood. So we have two sets of two different populations, the one coming from Rusmap, for which we have these 4 data modalities, and from brain data, from the brain, and specifically from the frontal cortex. And also we have this data modality from blood, and then we analyze it then independently. So the first intention was to we already have a way to organize the subjects in, let's say, to have a better picture, a set that snapshots of, or a more continuous picture of how the disease develops, right? Now, we know that the disease is not only one. When we talk, or usually we hear about Alzheimer's or Parkinson, it's very, very often assumed that there's only one type of Alzheimer's, when in fact, we know there are different variants of the same disease, right? The same that happened with COVID. We have the Omicron and the other variant, the Delta, etc. The same happens for neurodegenerative disorders. So we have the same when we analyze atrophy brain for person with Alzheimer's, we found that there are 3 or 4 different subtypes, and this subtype one have more atrophy, other ones have less atrophy, and it's more hippocampus related, etc. So we assume, or we believe that the same happens at the molecular level. So then we extended the model to integrate these different layers of molecular information in a way that would allow us not only to align the subjects, like in terms of continuity across the disease, the development of the disease, but also that we identify sub-trajectories, each of them corresponding with different disease variants or putative subtypes. And that's what that model did. So we used an iterative machine learning algorithm that first did the dimensionality reduction, and then it fused the different layers of information, trying to keep what is complementary across them. And then in that multi-dimensional space, identified concatenations of subjects that were aligned, having similar characteristics. And then we finish with always with, again, a pseudo-time measure from 0 to 1 that will be reflecting how advancing someone in terms of developing the clinical dementia, and with, for each subject or participant, a subtype assignation and the probability to belong to one subtype or the other one. And we go back to the subtypes after. We verified again that the pseudo-time, similar than the previous case, predicted neuropathological alterations like level of neuritic plaques, amyloid deposition, atherosclerosis, hippocampal sclerosis, not only the proteins deposition. And we also found that it significantly associated with hippocampal sclerosis, vascular infarcts, atherosclerosis, tangles in the hippocampus. And these associations were considering the redundancy. So we did a lasso regression. So what we have here is how these different macroscopic brain alterations associate with what the molecular integrated or aggregated molecular score of disease advance says, right? Yes, actually, we cannot say that these are the regions contributing to that molecular index, or that the molecular index contributes to this, but that when we see, let's say, when we identify these macroscopic brain alterations, they are somehow already reflected at the molecular level to the extent that the pseudotime or the multi-omics disease progression score reflects. And importantly, we can go back, or we went back again to see what specific molecular factors are contributing the most to predict the advance of the disease. Not only at the level of the RNA, but also at the considering proteomics, methylation, gene methylation, metabolites. And we found, again, factors like associated with morphogenesis, antibody recognition of tau filaments, or associated with previously identified to be related with clinical deterioration in Alzheimer's, with metabolism, with plasticity, with cardio and neuroprotective effects. It's something very interesting and important that we didn't find only one factor, that will be the top, explaining all the variants. We always find multiple of them across different molecular levels. And that's a point of support to the fact that the disease, or these diseases, are highly multifactorial. So, and why it supports why we need to consider multiple interactions and not only one dominant way of analyzing, or believing on one of them. Let's say most of the hypotheses I mentioned before are good to some extent and fail to some extent. And for me, one of the reasons is that each of the univariate hypotheses is somehow predetermined to fail because cannot cover the multi-dimensional problem or complexity of these disorders. Then we analyze how we detected 3 major subtypes of Alzheimer's disease considered the multi-omics data. They were not different in terms of proportion between females and males. Well, we see here that the females presented a higher percent, but the females were more common in the study. So at the end, if we control by the number of females and men in the study, both were present in the 3 subtypes. Also, they capture very well. So they have subjects that are intermediary disease stage, subjects that are more advanced stage. And we estimate that 2 subtypes were more affected than another subtypes in terms of episodic memory, and that the subtype number 1 was declining faster in time, in perceptual speed, episodic memory, semantic memory, global recognition than the other subtypes. So it means that the subtypes identified at the molecular level, so these were subgroup of patients or participants that have a different behavior in terms of molecular alterations across the disease progression. They were associated with different clinical manifestations too. Some of them have like what we call like a more aggressive version of the disease than the others, or a faster progression than the others, and that was reflected at the clinical level too. And then we went back to see how these subtypes were different across the different molecular levels and neuropathological levels. And what we have here are like just the results of how they differ in terms of gene expression, epigenetics, metabolites, and brain phenotypes. So as you can see, the subtypes have a lot of differences at the molecular level. So some persons have Alzheimer's with some molecular alterations, and others have Alzheimer's, similar clinical manifestation or relatively close, that just in the clinic will be called again with Alzheimer's disease. So there will be only one diagnosis, but there will be multiple ways of arriving to that diagnosis. And that not only happens at the molecular level, but we can see that in the bottom part, on the bottom graph, when we have different brain phenotype of macroscopic alterations associated with the different subtypes. And that reflects that the persons with the same diagnosis also can have different macroscopic brain alterations. Some brain regions will be obviously or at the macroscopic level altering one of these persons and not in the other one. And the same happens with amyloid and tau. So we could see that some subtypes have more accumulation of amyloid and tau than the others, or vascular infarct or hippocampal sclerosis than the others. And that's very important because it means that AD heterogeneity cannot be entirely captured or explained by neuropathological factors, clinical severity, or demographics. So the disease is a lot more complex. If we go still today in Mayo conference, we see a lot of effort focusing on only one biological factor, or only, let's say, or a small combination of demographics to explain all the heterogeneity and the whole disease spectrum. And that could provide some interesting results, but at the end, it's like trying to be explaining a multi-dimensional, highly dimensional space with only one or two variables. And we will fail, to explain the causal mechanisms of a complex disorder in that way. And if we fail to explain or to understand the disorder, of course, we're condemned, with a high probability to fail treating that disorder too. We also said it like for the initial pseudotime coming from gene expression, that the multi-omics molecular pseudotime of disease progression was significantly predicting memory performance and language performance. And in the vivo data is also associated with neuropathological alterations in the brain. And this result is very important. It's showing or supporting again that with a simple blood, well, it's not that simple, but a relatively minimally invasive extraction of blood, we can get robust diagnostics of what is happening in the brains of the person, and in terms of neuropathological effects, but also in terms of behavioral and cognitive capacity. And remember that in the future of the clinic, like in not all the places where we have access to expensive MRIs or expensive scanners, right? A PET, an amyloid or tau PET evaluation, I think costs over $1,000. While a blood evaluation is not that high. So it not only requires only blood extraction and the place that we do the screening and quantification of the genes and the proteomic data, but also it's a lot more accessible, a lot more easy to apply in the daily clinic than to use all the time of more complex equipment. I'm not saying that the more complex and special equipment is not necessarily, all the contrary, it's very necessary to understand the disease mechanisms and processes. But that if we expect to treat these disorders at massive scale, we also need to find simple, relatively simple, direct, minimally invasive ways to evaluate the patients. And then, in addition, from the blood data, we also found 3 minor subtypes of Alzheimer's, and one of them, again, advancing faster than the others. And here's when the things get relatively more interesting. Then we try to compare if the subtypes obtained from the brain, let's say the Alzheimer's subtype obtained with brain data, in one population, we have common information from those obtained with blood data in another population. And then when we did, we extrapolated the subtype from, remember, they are not the same subjects. We cannot just calculate correlation or mutual information between them. They are different type of data, so I will not expect to find a 1-1 correspondency. I will just expect to find that the classification system that we obtained for the brain will somehow contain information also represented on the classification system obtained from the blood data. So what we did was we extrapolated using neuropathological information, the subtype from the brain, to the subjects that only have blood data and the image information, etc. I will not enter too much into detail, but at the end we finished with, for the ANI participants, those that are alive, with 2 classification systems or 2 subtypes for each, one coming from the independent brain data, one coming from the own brain data, and we calculated the mutual information between these systems, and we observed that there is significantly overlap in comparison with random classification system, which means that also the blood classification system or the blood evaluation that we did is reflecting the ground truth that is the post-mortem brain evaluations. And again, supports how useful this could be for clinical, fast clinical daily applications. To get this to, at some point, to apply it in the real clinic, we are doing our analysis, which include independent cross-validation in all 3 different cohorts. 2 of them are from McGill University, one collected by us, one other collected by Pedro Rosaneto, which is clinic, and the other one is a Canadian study in aging and neurodegeneration, which I have also collected molecular information across different neurodegenerative disorders. So those are empirical models. We can get, they are useful, so they can be useful for the clinic. We also get like an explanation to some extent of which biological processes are more relevant for predicting disease progression, but we don't get an explanation of the disease. For that, and I presented this model before, we have also worked or defined using differential equation modeling these frameworks in which we try to analyze how the different biological factors interact to cause a given alterations that could be of interest, and how those alterations are projected to cognitive, the individual cognitive performance and clinical states. Essentially, this is the same model that I presented ago, which proposed that changing a given biological factor in a given region are a function of local interactions between other biological factors, plus incoming effects from other brain regions that propagate through vascular networks, functional and vascular anatomical functional networks, plus a standard impulse if the person is taking treatments or not, that can be translated to differential equations. I will skip that part. And then in practice, what we do is that for each participant, we have different imaging modalities measured at different time points. And this allows us, in combination with a specific optimization techniques, to solve the differential equations for each subject and to estimate the ways in which tau, amyloid, cerebral blood flow, amyloid, so the imaging derived factors interrelate in a direct way, controlled by indirect effects, and how those interactions are, those propagation across the brain impacts the cognitive level. Once that we have that, then we can see first how the model fit the real data, how the model fit the data. And what we have here is how the model reproduces amyloid, tau, and brain matter atrophy in the brain of a person for almost a decade. So we can use the model. Once that we feed the data, we can make interpolations and extrapolations to have a more continuous picture of how the brain of that person was changing in time, while considering the multifactorial interactions that might be happening. So essentially, we verify that the model can explain very well the data of the patient, but also that if we leave some time points out, the model can predict with high accuracy how will be the multifactorial brain of that person in the future, so that we can predict, let's say, up to 3-5 years after, what will be the level of amyloid accumulation in that person, how it will continue accumulating, or how it was in the past. And then this can be used also to make predictions in the clinic and to provide estimation of how accentuated, how much advanced will be the person in the future. It can also be used, I guess this is interesting, but I guess you will, well, okay, in the clinic, how this can be helping the people in practice, yeah, because at the end, if we give to a person how the brain, his or her brain will be in 10 years, what the person can do with that, I will not know what to do myself, right? So how can we use this to infer optimal or close to optimal therapeutic interventions at the individual level? So if we say the same model, remember that the model depends also on the standard inputs that a person can have, right? If he's doing physical exercise, what kind of diet, what kind of therapy he's having. If we invert the equation of the model and we pass to the left side of the equation, a standard inputs component, and to the right side, all the other components. Once that we fit the individual data, let's say once that we have a model, individual model that captures how the different biological factors are interacting in a given time window, let's say for 5 years or 6 years of a person, then we can use it to make simulations or in silico projections of where the brain and the cognitive features of that person will go if we make a small change in the brain, right? So, and we did that. So we defined something that we call multifactorial intervention fingerprint. That is no more than estimations of how efficient would be to target or to stop brain deformations by targeting the specific biological factors that we are studying. So imagine that you say, okay, I already have the differential equation for myself. Now I will, in the computer, simulate what will happen if I reduce amyloid in my brain. And then you will have projections, future projections of what will happen and how that changed the cognitive states or not. So you will have that in the continuous space. And then I will simulate what will happen. I don't know, if I increase my brain metabolism or decrease it, or decrease my brain activity. And again, you get estimations of how close that gets you to be normal or not, or how much deformation you need to introduce, right? And then we estimated individual measures or values of how effective they would be for all the patients that we have. And then we found different types of patients with the same diagnosis. For example, we found the patient number 1, let's say, that according to the model, vascular or a metabolic intervention will be the most effective. And for that person in the clinic, what the doctor could suggest is to do physical exercise, ketogenic diet, changing the diet, being more active, etc., or having interventions, drug compounds that could improve the integrity or the functionality of the vascular system. For another kind of patient, we found that an anti-amyloid and anti-tau intervention will be more effective. And the treatments that are approved right now for Alzheimer's, the new ones, are exactly targeting more amyloid and tau. So that person could use those ones. And for other kind of patients, we found that none of the single target interventions will be effective because that person is probably too advanced or have a variant of the disease that is too multifactorial or highly multifactorial in comparison with the others. So for that one, what we suggest that they need a multifactorial intervention. So we can also simulate combined interventions. So we can, in the modeling, again, simulate what would be the effect of reducing amyloid and increasing and decreasing atrophy, or reducing amyloid and tau at the same time. And we did all those estimations. So in essence, that if we summarize that framework of that fingerprint model or approach, what we do is that we collect or obtain different data across different imaging modalities and time points for each person. And then we use the multifactorial causal model to estimate how the brain of that person is working, how the things are interacting, how they are spreading across the brain. And then we use control theory and brain simulations and model simulations to estimate the energy or deformation that will be required to bring that person to a desired state, which could be a normal clinical state or could be just being stable. And then this allows us to estimate these numerical values reflecting how efficient what type of treatment will be in comparison with other one. We can also use the genomic or transcriptomic data from blood, for example, to know which are the genes associated with each specific treatment that the person, the model is suggesting. And actually that's what I have here. So we use imaging data to divide the persons according to their treatment mix or the primary treatment mix. And each cycle here or each ring corresponds to subjects subgroup according to different treatment needs. For example, the first one corresponds to those needing vascular intervention, then amyloid, then tau, etc., until structural interventions. And what we have in black are which corresponds to a specific molecular pathways that are altered on those that need each kind of intervention. So we can go from getting an estimate of what intervention will be needed at the individual level to an idea of what molecular features or genes are altered, specifically altered on each person, which can allow to understand why the person needs that specific treatment. Something interesting that we found is that the person with vascular and amyloid, so that the model suggests that have vascular amyloid interventions, they were the ones having less molecular alterations at the peripheral level, which again supports the idea that this could be very amyloid and tau, vascular and amyloid features could be very early on the whole disease process because they have less molecular alterations, right, in the blood. And then those needing a structural brain intervention are the ones that have more, a lot of molecular pathways altered in the periphery. We see that at the point that we have a lot of brain atrophy or structural damage, or we have a lot of other biological factors altered even in our periphery. Of course, we cannot yet say, okay, you need this specific treatment, like this specific drug, etc. That's something that we are working, but it's not here yet. And there are other challenges implied, right? Like for those that they need more in structural intervention, what does that mean? So it means to have to control neurogenesis, right? We don't have a clear way. I mean, on the other side, on the clinical, at the pharma level, there are no clear ways to control neurogenesis in the brain. So I guess this is informative and can be useful in many ways. So if someone needs a neurogenesis and why giving the person an anti-amyloid, an anti-tau, and in fact, the thing that will benefit more the person is something that doesn't even exist, and those other treatments might not be effective. And this could work in the other direction, right? But we have to consider this in combination with how feasible are those treatments or not. If the model gives me that someone needs whole brain neurogenesis, I know that treatment doesn't exist, so maybe that person, the doctor, will decide to give something that already exists, even if it's not the optimal. And we did this not only in the context of Alzheimer's disease, then we tried to see how it could work for people that are already taking drugs. So we applied it in the context of Parkinson. We used the data from the PPNI database. It's another major study, in this case associated with Parkinson. A big part of the population in that study have received levodopa or variants of levodopa. That is the most commonly used drug for Parkinson's disease. And then we evaluated how well this person was responding to that treatment or not. Then we applied the model separately, and we identified that the model could predict or could explain why someone would respond or not to levodopa, or to what extent someone will be a responder or not of levodopa. And in addition, we did the same molecular analysis from the blood, and we found that the genes and molecular pathways that, according to the model, were relevant to respond to that treatment were the same that were differentiating the people that were responders and not responding in the treatment on the other side. So there was a high overlap between what the model was suggesting, like the computational mathematical differential equation model from only imaging data, and what we observed from the peripheral molecular and clinical side. And we are applying other ways, or we are using other clinical trials to verify that the model's predictions can actually give valuable information in terms of who will respond or not to a given treatment. Until there, I said, let's see, for the mechanistic model, almost everything is coming from neuroimaging data. And we use peripheral information, with blood expression and blood transcriptomics and genotyping, but we haven't yet incorporated molecular information within the differential equations of the models. So that's what we did, and specifically Quadri Adewale, PhD candidate in my lab, did in the last year. So he expanded the model's equations to decompose the multifactorial interactions that were occurring before only at the macroscopic level, and into gene interaction or genetic interactions that will be controlling those interactions. So imagine if we say, now we want to not only estimate how, for a given person, amyloid is interacting with the cerebral flow, but also want to see which genes are controlling those interactions, right? We want to detect that. And that's very important for multiple reasons. So while we expanded the equations, we used a specific data to estimate that. And that allows us to finish with this kind of broad map, in which what we have is, so on the inner cycle, we have the biological factor of interest. Let's focus here on tau. What we have is tau, and then the second ring, what we have is how all the biological factors interact with tau. So tau is mostly affected by itself. So by accumulation in cells, seems to be promoting more accumulation of tau. But then we have on the standard cycle, which genes are actually mediating the effect of tau on itself, right? Or the effect of atrophy or glucose metabolism on tau. So it means that if you, let's say, you're planning to develop treatment or a drug to control tau propagation, tau accumulation and propagation in the brain of one person, then you can go to those genes that are in the standard range, and you already have a very reduced pool of genes that, according to the model, have a controlling effect of what happens to on the other side. Not only a correlation, indirect effect, which is what happens for many, when we see, let's say, in many other genetic studies or other ways to analyze genetic data, we finish having just genes that correlate with, I don't know, amyloid, atrophy, diagnosis, etc. In this case, these are genes that control the multifactorial interactions between multiple biological factors and the one that we could care about. And we can do the same with brain atrophy or functional activity. If we go to functional activity or blood flow, we have the genes that modulate the interaction between glucose metabolism and blood flow. And if we want, for some reason, to move the vascular system from one, say, in one direction, we should focus then on those genes. And we have numbers. So when I say focus on the genes, it's not in a binary or a yes or no way. We have weights associated that reflect how much we need to focus on one gene in comparison with the other one. And another PhD candidate in the lab, Ahmed Khan, who will be presenting today, extended this also to have to consider neuroreceptors information or neuroreceptor densities across the whole brain. And this was just done in collaboration with Carl Siles and Nicola Palomero from the Jewish Institute in Germany. As Ahmed will explain, we again decomposed the multifactorial interactions between these different biological factors as a function of the neuroreceptor densities across the different areas. And this again, similarly to the gene expression case, offers like this roadmap in which we have which specific neuroreceptors interact and control the interactions between the basic biological factors that we are considering and other biological factors. Essentially, they also offer potential ways to go from the molecular level to target the molecular level to cause a desired effect at the macroscopic brain level. We are also using neuronal activity simulator decoders to understand better the disease process. And this is another line of research in my lab. Until now, we have considered different biological factors derived from neuroimaging data, I would say in time, but we haven't tried to model, we haven't modeled neuronal activity, right? Fast fluctuations in neuronal activity and how that associates with the neuropathological factors or cellular factors and molecular change in the disease. So that's exactly what Lázaro Sanchez is a PhD candidate in the lab have done in the last 3 years. So we have used neuromass models that essentially model how the neurons, how neuron activity is generated at the regional level and with input from other brain regions. And we know which regions are connected thanks to the anatomical networks. To know how that activity is affected by amyloid, tau, the interaction between amyloid, tau, or other relevant biological factors that are quantified in that region. So essentially what we do is that we assume that neuronal activity or specific factors controlling neuronal activity, like could be like, for example, the threshold of the synaptic propagation depends on amyloid, tau, etc. And then we estimate model parameters, individual parameters that should or that are associated to the way that these different neuropathological components have on neuronal activity. Once that we have that, so we do that, the estimation is done simulating, again, based on simulations, we simulate data, trying to maximize the parameters or trying to maximize the similarity between simulated and real data. And in that way, we optimize the model parameters. And based on that, then we have observed that the identified models reproduce well electrophysiological patterns already observing Alzheimer's disease, and that it also the model parameters associate significantly with cognitive performance of the subjects and also with peripheral biomarkers that have been proposed recently. This is still under development. We have an initial paper, an article submitted, and we have working on new extensions, and we continue to enrich these models to other kinds of biological data. So it goes from information at the, in this case, neuropathological level, but can also be molecular level, to estimation of how that affects neural activity, continuous fast neural activity, and projection of that to the individual brain. Again, this can be used for treatment selection, so we can do simulation. Once that the individual data, we have estimation for the individual data, we can simulate in silico again what would be the effects at the neural activity level of reducing amyloid, tau, or the interaction. And finally, I will briefly talk about how we can take this to another level, and now more still deeper at the molecular, deeper towards a molecular characterization to enrich bottom-up models. I talked before about gene expression, proteins, etc., but there are other modalities of molecular layers that are relatively new and that have shown in other contexts to be very informative. So normally when we have gene expression data, it is like taking a snapshot of the concentration of activity level of a gene at a given time point. If we have that, let's say, for the postmortem brain, it means that what we have is like how more or less the genes were activated at the one time point. But that doesn't really reflect the dynamics of the gene, right? And so imagine if we could estimate also how that gene was changing at the time of evaluation. Now we give an idea not only of the biological processes that were altered or different at the time point, but also an idea of which biological processes were changing faster. So it can even provide a better biological explanations of the whole process. So we have used a new metric to estimate gene expression or RNA velocity, and this is we need single cell data. We have applied it to the Alzheimer's disease case, and essentially it allows to estimate or to infer the directionality and velocity of the molecular change at the given tissue. And that's very important because that will allow in the near future to not only include in the models information of the concentrations of a given gene or protein or metabolite, but also how they were changing in time and what will be the future value of which could be the future direction. And in other terms, it provides information that cannot be seen with the snapshots of the data, and that also highlights or allows to identify all the molecular processes that we will usually miss because we don't look at the dynamics of the molecular information. We have identified other with differential RNA velocity, other molecular factors and pathways that have been missed traditionally because we have been traditionally only looking to what happens at a given time point. It's like imagine if you have a curve, not only having the value of one position, but also the slope of that specific, or the derivative on that specific position. So you have an idea of which will go up or down in the future. All these models, as I mentioned before, are available or will be available for the last ones in the user-friendly interface that we share in the lab, and that will allow us to go from incorporation of the molecular, multimodal, neuroimaging, therapeutic data to visualization in the brain, and it also allows us to create videos of how the brain is changing in time to make predictions of what will be the level of alteration in the future, or to simulate effects of specific interventions. That software have, again, both the mechanistic modeling I have talked about and also the machine learning. And we believe that there are no reasons to work on only one side of both. There are advantages and disadvantages of each stream. And what we try to do is to move towards a middle ground or to create a bridge between both modalities or main strategies. With that, I want to thank again the organizers and the audience for attending and the members of my lab from the institution. Thank you.